Oximetry is a non-invasive medical technique used to measure the oxygen saturation level in a person's blood. It involves the use of a small device called a pulse oximeter that is typically placed on a finger or earlobe. The pulse oximeter uses light absorption to determine the percentage of oxygenated hemoglobin in the blood. This information can be crucial in assessing the respiratory and cardiovascular health of a patient, as well as monitoring the effectiveness of oxygen therapy. Oximetry is commonly used in medical settings, such as hospitals, clinics, and during surgeries, but portable pulse oximeters are also available for home use. Oximetry applications refer to the various uses and benefits of measuring oxygen levels in the blood using an oximeter device. Oximetry is commonly used in healthcare settings to monitor and assess the oxygen saturation levels of patients, particularly those with respiratory or cardiovascular conditions. It is also utilized during surgeries and intensive care to ensure adequate oxygenation. Additionally, oximetry has found applications in sports and fitness monitoring, allowing athletes to track their oxygen levels and optimize their training. Furthermore, oximetry is increasingly being integrated into wearable devices and mobile apps, enabling individuals to monitor their oxygen saturation levels at home and receive early indications of potential health issues. Overall, oximetry applications have revolutionized patient care and wellness monitoring by providing quick and non-invasive measurements of oxygen levels in the blood. In vitro oximetry is a technique used to measure the oxygen saturation levels in a sample outside of the body, typically in a laboratory setting. This method involves using specialized equipment that utilizes light absorption to determine the amount of oxygen bound to hemoglobin molecules. By analyzing the color changes in the sample, the oximeter can calculate the oxygen saturation levels, providing valuable information about tissue oxygenation and the efficiency of gas exchange. In vitro oximetry is commonly used in medical research and diagnostics to study oxygen levels in blood samples, as well as in quality control processes for medical devices and pharmaceutical products. Transmission oximetry is a non-invasive method used to measure the oxygen saturation levels in a person's blood. It involves the use of a sensor that emits light at different wavelengths, usually near infrared light, and measures the amount of light that passes through the body. By comparing the intensity of the transmitted light to the original light source, transmission oximetry can determine the percentage of oxygenated hemoglobin in the blood. This technique is commonly used in medical settings to monitor oxygen levels in patients, particularly those with respiratory or cardiovascular conditions. The spectral characteristics of blood refer to the unique way in which blood interacts with light across different wavelengths. Blood has distinct absorption and scattering properties that can be measured using spectroscopy techniques. The absorption spectrum of blood is influenced by the presence of various chromophores, such as hemoglobin, which absorb light differently depending on their oxygenation state. This allows for the measurement of parameters like oxygen saturation and concentration of hemoglobin in the blood. Additionally, the scattering of light in blood is influenced by the size and concentration of red blood cells, providing information about cellular morphology and density. By analyzing the spectral characteristics of blood, researchers and healthcare professionals can gain valuable insights into various physiological conditions and monitor changes in blood composition. In the field of biochemistry, the molecular extinction coefficient is a measure of how much a particular molecule absorbs light at different wavelengths. For hemoglobin and oxyhemoglobin, the extinction coefficient can be plotted against the wavelength of light to reveal their unique absorption patterns. This information is crucial in various applications, such as determining the concentration of these molecules in a sample or studying their behavior in different environments. By understanding the absorption characteristics of hemoglobin and oxyhemoglobin across different wavelengths, researchers can gain valuable insights into their structural and functional properties, leading to advancements in areas like medical diagnostics and therapeutics. Reflection oximetry is a non-invasive medical technique used to measure the oxygen saturation levels in the blood. It involves shining a light onto a specific part of the body, such as a finger or earlobe, and then detecting the amount of light that is reflected back. By analyzing the color and intensity of the reflected light, the device can determine the percentage of hemoglobin in the blood that is carrying oxygen. This information is critical in monitoring the respiratory and circulatory health of patients, particularly those with conditions such as lung diseases or heart problems. Reflection oximetry is widely used in healthcare settings, providing a quick and accurate assessment of oxygenation levels without the need for invasive procedures. The diagram illustrates the arrangements of the reflection oximeter. In vivo oximetry is a non-invasive technique used to measure the oxygen saturation levels in living tissues and organs. It involves the use of specialized devices, such as pulse oximeters, which emit light and measure the amount of oxygen carried by the hemoglobin in the blood. This technique is commonly used in medical settings to monitor oxygen levels during surgeries, in intensive care units, and for patients with respiratory or cardiovascular conditions. In vivo oximetry provides valuable information about tissue oxygenation, helping doctors make informed decisions about patient care and treatment. An ear oximeter is a medical device used to measure the oxygen saturation level in the blood. It is designed to be placed on the earlobe and uses infrared light to analyze the amount of oxygen carried by the hemoglobin in the blood. By providing a quick and non-invasive way to monitor oxygen levels, ear oximeters are commonly used in hospitals, clinics, and even at home for patients with respiratory conditions such as asthma or chronic obstructive pulmonary disease, COPD. The device is also useful for athletes and individuals at high altitudes to track their oxygen levels during physical activity or altitude changes. Overall, ear oximeters play a crucial role in assessing the respiratory health and well-being of individuals in various settings. The images show the block diagram of the ear oximeter. A pulse oximeter is a small portable device that measures the oxygen saturation level in a person's blood. It does this by emitting a beam of light through the fingertip or earlobe and detecting the amount of light that is absorbed by the oxygen-carrying hemoglobin in the blood. This information is then used to determine the percentage of hemoglobin that is saturated with oxygen. Pulse oximeters are commonly used in medical settings, such as hospitals and clinics, to monitor patients' oxygen levels and quickly detect any potential respiratory issues or oxygen deficiencies. They are also increasingly being used by individuals at home to monitor their own oxygen levels, especially for those with respiratory conditions or during physical activity at high altitudes. A pulse oximeter is a medical device that measures the oxygen saturation level in the blood. It consists of several key components. Firstly, there is a probe or sensor that is typically placed on a finger or earlobe to detect the oxygen levels. This probe emits light at two different wavelengths, typically red and infrared, and measures the amount of light that is absorbed by the blood. Secondly, there is a microprocessor that processes the data received from the probe and calculates the oxygen saturation level. This microprocessor also controls the display and other functionalities of the pulse oximeter. Lastly, There is a display screen that shows the oxygen saturation level, and sometimes the heart rate as well. Some pulse oximeters may also have additional features such as alarms, memory storage, and connectivity options. Overall, these components work together to provide a non-invasive and convenient way to monitor oxygen levels in the body. Figures show the typical pulsing of red and infrared light emitting diodes by a pulse oximeter. Figure illustrates the infrared light absorption in the finger. Analog signal processing by pulse oximetry. For the measurement of oxygen saturation level of blood in localized areas of oxygen-deprived tissues on the limbs, head and torso, a skin reflectance oximeter can be employed. The instrument basically depends on monitoring backscattered light from living tissue in two wavelengths. The backscattered light data is then used for the in vivo determination of the blood's relative oxygen saturation. Schematic diagram of the reflectance oximeter sensor. An intravascular oximeter is a medical device used to measure the oxygen saturation levels in the blood. It is inserted directly into a blood vessel, typically in the central venous system, allowing for accurate and continuous monitoring of oxygen levels. The device uses a sensor that emits light and measures the amount of light absorbed by the blood, providing real-time data on the oxygenation status. This information is crucial in critical care settings, such as intensive care units or during surgeries, as it helps healthcare professionals assess the patient's oxygenation levels and make informed decisions about their treatment.